Hi, I'm Avia, and I'm a PhD student from a Sephardi's lab at the Weizmann Institute of Science. I'm working on stress sensing and sulfate regulation in marine diatoms. Diatoms, as other phytoplankton, are constantly exposed to stress conditions in their environment, and they need to sense and respond accordingly. They can do that by using reactive oxygen species, or in short, ROS, that integrate information coming both from within the cell and from outside of the cell. Worms are constantly produced as byproducts of aerobic metabolism, but they can also greatly accumulate in response to biotic or abiotic stress conditions or due to signaling cascades that lead to their production. Depending on the specific conditions, these ROS signals can lead to either stress acclimation or, under harsh conditions, to programmed cell death. Programmed cell death, or in short PCD, is a regulated form of cell death that was suggested to be involved in the high turnover rates of phytoplankton and as a major mortality mechanism during bloom demise. However, we know very little about the molecular machinery that regulates and executes it, and phytoplankton, as other unicellular organisms, are lacking most of the canonical PCD machinery known from other systems. We also don't know what are the downstream effects of PCD regarding community structure and carbon fluxes compared to other modes of cell death. And we also don't know how PCD was able to evolve in unicellular organisms despite being a little trait, although there are many theories that may explain it. When I started the project, I was interested in understanding how can some cells survive stressful conditions that lead to the killing of most of the population. And if you think about it in the context of the bloom, during the demise, most of the population dies, but some cells have to survive in order to serve as a seed for the next bloom. I started investigating this question by characterizing heterogeneity in Phaeodactylum tricornutum populations in response to oxidative stress that leads to partial killing of the population. And I used a genetic reporter that represents the oxidative stress in the chloroplast. Under control treatment on the left side, most cells maintain a reduced state in the chloroplast, represented by this purple-blue color. But when the cells were treated with hydrogen peroxide, some cells became completely white, as depicted by the red arrow, representing an oxidized state in the chloroplast, while others were able to maintain a more reduced state and even reduce and acclimate later on. So this exposed two distinct subpopulations that emerged following oxidative stress. And indeed, the cells that were initially just 40 minutes after treatment were oxidized in the chloroplast also died 24 hours later, while cells that were able to maintain a more reduced state were more resilient to the stress and were able to survive. And these cells are coming exactly from the same culture, from the same genetic background. So next, I wanted to use this system in order to identify genes that are involved in sulfate regulation. I treated the cells with the same treatment and then sorted the reduced resilient subpopulation and the oxidized sensitive subpopulation, as well as the control untreated cells, and performed transcriptome analysis of these sorted subpopulations. Indeed, despite coming from exactly the same culture and the same conditions, the same treatment, this subpopulation exhibited completely different transcriptional states, and I could detect clusters of genes that are upregulated specifically in the resilient cells or specifically in the cells that are going to die in the sensitive subpopulation. And this led to some interesting candidates that are under an ongoing investigation. And I intend to generate knockout mutants in these candidates using the CRISPR-Cas9 system. However, at the beginning, I had hundreds of candidates and I needed a way to narrow down the list to only a few for functional characterization. For that aim, I utilized an orthologous system in the green alga Chlamydomonas, in which a large-scale library of mutants was generated and was phenotyped in different conditions. So by combining a phenotypic data from an orthologous system and a transcriptional data from PT, I was able to narrow down the list to genes that seem to be more relevant and likely to be conserved. I also intend to use this transcriptional data set in order to identify sensitive stress biomarkers that may be utilized in natural diatom populations. With that, I would like to thank you for listening and thank all the people who are involved in this project. And feel free to contact me later if you want to hear some more.